Our next stop takes us some kilometers north to the city of Magdeburg, where we will visit the University Otto-von-Guericke. Let's have a look at the Laboratory for Image-Guided Therapy, which will be presented by Dr. Alfredo Illanes. So enjoy Alfredo's video. It was done very short time, just last week. So but I hope you like it.
Hello everyone, I am Dr. Alfredo Illanes and we are here at the INKA HealthTec Innovation Lab here at the medical faculty at the Otto-von-Guericke University in Magdeburg. So this lab was initiated by Professor Michael Friebe, and what we do is innovation, device innovation for minimal invasive procedures and health monitoring. So the lab consists of three main parts. I will show you the first part, that is the prototyping and electronic room. Yeah, so we have here Charani and Thomas. They are working on one of our intelligent devices. So here you have a orthopedic palpator that is connected to a vibroacoustic sensing device. What this sensing device does is to listen to the tool when this tool touch tissues. So now what they are doing is to extract parameters from the audio that are related with parameters of the material. So if you follow me here, so here we arrive to the electronic lab, and here we have Anna that she's working on sensing in what is the name of this? Implants, yeah, orthopedic implants. So the idea is to place sensors in these orthopedic implants to be able to monitor them from outside the body. I hope that I say it good. Yeah. So we go to the second part of this lab. The second part of this lab is the creation lab, and here people try to be innovative. They create their processes. So we have Arca here. He developed embedded system, we can say, yeah, for surgical audio guidance. And we have Holger and Vicky that they are working on a injector, MRI injector, very low in cost. Yeah, this is the idea. So if we continue here, we enter to the third part of the lab, that is the medical experimental part. We go here with Axel. So what Axel is doing here, he's inserting a vascular endoscope, so it's really tiny. He's going inside the phantom of an artery, and what he's trying to search are anomalies inside the inner wall of the artery with this device. Finally, we are here with Nazila, and this is very important because this is the project that I will talk later. So Nazila, what she is doing is monitoring needles. Yeah, so we place an audio sensing device on the needle. The needle starts to break tissues, and we can identify these events to provide us feedback to the surgeons. So yes, so every one of you are welcome here to this lab, to the INKA lab. We are searching for collaboration, and see you soon in my presentation. Thank you very much.
Okay, so I can share my presentation, yeah? Okay, so I will share here. I will share the audio. The audio here is very important. Can you see?
Yes.
I have a problem here. And now, yes. Okay, so hello everyone. So first of all, thanks a lot for this invitation. I am Dr. Alfredo Illanes. I work at this INKA lab since end of 2015. And what I will present you in this presentation is one of our main projects here at INKA today, that is the Surgical Audio Guidance Project, so SURAG project. So that means how can we guide medical interventional tools using acoustic emission. Yeah, so this is a project that starts in its research phase in 2015. And since 1 year and a half, we are in a research transfer project financed by Exist Fortune Transfer here in Germany. I have here. So yeah, but let's make first an additional brief introduction about INKA. So we are located at the University Clinicum in Magdeburg. So we have our offices and our laboratory here at the hospital. So this is very good for us because we are next to the surgeons that are our main collaborators. So INKA is highly focused on applied research and development of medical device. And this is because of the background and experience of Professor Michael Friebe that initiated INKA. So in fact, Professor Michael Friebe, he comes from the industry. You'll see he's a innovator, investor, and scientist. He has started already more than 15 companies, 5 as major shareholder. Even he was elected Germany's Business Angel of the Year 2011. He has filed more than 80 patents. So you can see a little the vision of Professor Friebe for the work that we are doing today. So our team is composed today by 9 PhD students, 2 senior researchers, one of them is me. And we have between our master student, HIVIs, we have around 10 different nations and working in different fields as mechanic, informatic, electronic, material, data processing, like in my case. So some numbers since INKA was created in 2015 and until today, 2021. So we have published more than 260 publications in these years. We are very IP focused, so we have filed 37 patents. We have won already 8 innovation awards, and that's something that is very important. From INKA, already 4 startups have been funded. Yeah. And today we have 1 research transfer exist project, and we have this INKA HealthTec Innovation Lab. So this is what I was showing, or where I was during the video. So it's here you can see the INKA HealthTec Innovation Lab. So as I showed you, we have a creation room, yeah, and then a medical experiment lab with different equipment. So you have the ultrasound devices, endoscopic tower, optical tracking system, etc. And the innovation lab counts also with an electronic lab, with a prototyping lab, and also with an evaluation lab. As I told you before, we have won many innovation prizes since the creation of this lab. For example, 3 Hugo Juncker Prize here in Sachsen-Anhalt, 2 Science for Life, and one IQ Innovation Prize, and also prizes of best paper in different international conferences. So here's how we work. Yeah, you can see our methodology. So first of all, together with our surgeons, since this is very important to be here at the hospital, we identify clinical needs, we invent solution and business concept for these clinical needs, and then we implement them via, for example, proof of principles, research cooperation, and research transfer. This is why cooperation is crucial for us here. And this results usually in intellectual property publications and startup venture creation. This is our partnership today in terms of university. For example, we have a very good partnership with IIT Kharagpur in India. I was there even some years ago, 1 month. University of Porto, TU München here in Germany, QUT in Australia, Universidad Rey Juan Carlos in Spain, and also in Chile, yeah, because I'm Chilean, so we have to have some cooperation with Chile. Hospital, mainly we have cooperations, okay, as here in the hospital in Magdeburg, but also with the Clinicum Recht de Nizar at the TUM, but also with the Universität Klinikum in Ulm. Yeah. And during this 6 years, we have an active collaboration with industry. This is very important for us. So we have made projects, for example, with Brailab and General Electric, and currently we are closely working with Better Pharma. So I am at INKA, and I lead the data processing group that works in 3 main areas. The 3 main areas of my group are signal, image, and video processing, sensors development, and machine learning. And we apply this in 2 main topics. First of all, diagnostic and monitoring, and then guidance, navigation in minimal invasive surgery. So in terms of diagnosis and monitoring, you can see here, for example, projects, the ultrasound thyroid segmentation and nodule classification. We also do brain mechanical vibration assessment, yeah, for diseases. larynx cancer classification in contact endoscopy images, but also we put audio sensors in the knee joint for evaluation of the knee joints. In guidance and navigation, we work mainly on guiding laparoscopic and robotic tools using acoustic emission and ultrasound. And this is why what I will present you today is what the project, what is called surgical audio guidance. Again, what we do is by the acoustic emitted by the instrument when touches tissue, we can provide surgeons with significant information about about location of the instrument. Okay, so let's start here. What is the clinical motivation? So first of all, in open surgery, in open surgery, the surgeons can directly feel and touch tissue. Yeah, but when we are talking about minimally invasive surgery that is performed through some tiny incisions, the doctor cannot be in direct touch with tissues and organs. Why? Because there is an instrument, a long rod that is between the tissue and the hand of the doctor. So this means that the doctor will lose between 30 and 100% of haptic perception. When he loses 100% of haptic perception, for example, when we talk about robotic assisted surgery. So in minimally invasive procedures, surgeons usually should perform different type of tasks. Yeah, two important tasks are the insertion task, for example, usually with needles, and the palpation task. So examples of procedures where surgeons need to perform insertion tasks are laparoscopic access. So the creation of the access that is usually made with a Veress needle, with what we call an insufflation needle for creating the space of working for the laparoscopic surgery. We have also, for example, spinal injections, biopsies, and kidney stone removals that also the access is created with a needle. Examples of procedures where the surgeon needs to perform palpation tasks are arthroscopy. Yeah, for example, how we can assess knee stage, laparoscopy, and robotic assisted surgery. And all these procedures or tasks have a common point, is that they require haptic and targeting information. So several technologies have been proposed for support in this type of task. First of all, you have imaging systems as magnetic resonance, CT or ultrasound. Second, you have tracking system, for example, electromagnetic or optical. And finally, you have embedded sensors that they place at the interventional tools. So the value proposition of this solution is to provide to the surgeon navigation and haptic feedback information during the procedure that they are performing. For some procedures, I have to say that for some procedures, this has a great and enormous value, but for some other procedures with very simple workflow and setups, this can be over-dimensioned and add high additional cost. So the main drawback of these technologies is that they can be highly complex, costly, and sometimes they are cumbersome technologies. So this is why, I mean, I have to say I was formed in the field of signal processing, but I was working long time in vibratory analysis using acoustic emission, using accelerometers, and this is how we came to the idea 6 years ago. What if we can listen to the tool? Yeah. So what that mean? That means that when you have a rod, there are things on any instrument, and the tip of this rod enters in contact with tissue, at the point of contact, there is an audio wave that is generated. And this audio wave travels through the tool. And the important thing is it travels through the tool without the necessity of any active component. So we don't need any electronic component in the shaft or in the tip of the instrument. And what this audio that propagates through the tool, we can acquire from outside the body of the patient. And this is very, very important. What we receive is a very complex information, but with data processing, we can decode this information and we can provide feedback to surgeons. So here you can see a Veress needle Yeah, this is the needle that are used for accessing laparoscopy. You can see one of our sensing modules, and you can see how tiny tiny interactions of tissue with the needle propagates, and we can acquire outside in what we call in the proximal end of the tool. So if you think about, we can measure this, or we can listen to any audio, any medical tool. So you have to think on the potential application that this concept has. So we will go a little fast here. This is roughly how it works. So you have the instrument. We have a sensor module that send the information wireless to a processing unit, and in the processing unit we process the signal, we characterize the signal, we extract feature, we classify, and we decode information that can be useful for the surgeon. For example, the moment that the needle passed from one tissue to another one, the moment that we have a puncture or a perforation. We can also hear if a tissue is hard or is rough. Yeah, we can see underneath bony structure, and we can also hear the vessel presence inside the body. These are some of our prototypes. So we have full of prototypes, some very old, like this stethoscope one. This was our first prototype, and we have created prototype mainly for listening to needles, listening to laparoscopic and robotic assisted surgery tools, and also listening to guide wires. I will jump a little this. I will go directly to one of the video. I hope that you have the audio. The audio is very important. So here we have a needle going through chicken breast. So, okay, we cannot hear very good, but you can see exactly at the moment that we start to break this tissue and the moment that we start to go out. Now we are inserting the needle. Yeah. This is a needle that we are inserting in different type of fruits. These fruits are located in a gelatine phantom that is located 6 centimeter depth. And the more important here is not only that you can hear this, the most important here is that every type of tissue, when the needle breaks the tissue, generate a different signature in the signal. And what we are working today is how to characterize this signature in order to know what we are going through. This is another example. So this is a biopsy needle. We have there what we call a blind phantom. So it's a gelatine phantom that is with ink, like that surgeon. She's not surgeon, she's one of our colleagues here. But what we do is we invite surgeon to insert this needle because we want to know their perception. What we put inside are soft tissue. In this case, inside there is an olive, yeah, without the stone. And you will see the doctor, they don't feel when they are entering to this object, but you have to see how this object sound. Yeah, so there is the olive in the ultrasound image. Yeah, in the ultrasound image, you can roughly see when you are entering, but you will straight here. Then you can see the needle tip, the target, and the breaking of the tissue. Yeah, so tiny vibrations that are generated by the interaction between the needle tip and this soft tissue these vibrations create audio waves that transmit through the tool. This is what we are listening here. Here we have another one that is even a softer tissue. This is strawberry. You can see there's the strawberry inside. And again, you can see how these tiny interactions can be magnified. Yeah, using advanced signal processing algorithms. Okay, so also not only we can listen to insertion, but also we can listen to palpations. Yeah, this is a palpator that usually is used in orthopedic surgery, and this palpator, standard palpator, we connect to our device, to our sensing device, and you will see how sensitive it is and how a different tissue leave a different audio signature. Everything is isolated from outside, so we listen only to the tissue.
1, 2, 3.
And then what we can do is to analyze the signal, real time or offline, yeah, and we can extract characteristics from the signal, yeah. Characteristics, for example, in this case, the velocity of the audio wave. So we see that the ultrasound gel produces audio waves in the order of 9 oscillations per second. The foam between 20 oscillations per second, and the wood that is very hard produce more than 500 oscillations per second in the audio wave. I think that my time is finishing, but I will try to go. So what we are using this palpation information is for classifying cartilage states. So we are working together with the department, orthopedic department here, and what we've tried to do is we do histological analysis of the tissue, and we compare with the audio signature of the tissue to see the correlation between both. And our vision, this is our vision for this project. So what we want is to palpate different type of tissue. For example, we want to analyze. So we see what one tissue produce audio waves that are completely different than the other. And then we want to provide some information about how hard, soft, or how rough, smooth is the tissue. We have applied this concept also to robotic tools. Also, we can distinguish between different types of tissue. We can even hear pulsations, yeah, from the outside the body. Yeah. So for finishing fast, so this is the surgical audio team. So we have, so I am the person in signal processing. Thomas is the person who built all this hardware and sensor and electronic. Nasila is machine learning, and Moritz is the software developer. And we have our mentors, Professor Michael Friebe and Axel Böse, and we have also a big research network and clinical partner. So thank you very much. So if you have any question, do not hesitate. And I will be there today. Tomorrow will be Moritz there, but today I will be there at the virtual booth. So thanks a lot.
Dr. Illanes, thank you so much for this very in-depth presentation. Maybe there are some questions from the audience. There are. So there was first Dirk and afterwards Jürgen Drescher.
Thank you very much, Alfredo. That's a great presentation. Very nice. Just one question concerning the if your colleague Nasila, yes, that was her name, is doing the measurement and somebody else, like you, are doing a measurement. Isn't there a massive influence of the person doing these tests? And that the robotics you have just shown would be a better solution? Is it really reproducible? Just to out of interest.
This is a really important question, and it's true what you say. I mean, the user changed the characteristic of the audio. Yeah. So, for example, today, for arriving to be able to take the user out. Yeah, we have our robotic arm here, so we connect the device in the robotic arm. This gives us like a kind of baseline, but for taking out what the different user can produce, we need to have a full model of the device. Yeah, this is something complex, but what is interesting is that, for example, imagine that you are going with a needle and you cross layers. The events are always there. So when you do an event-based analysis, for example, a puncture always will be a puncture, doesn't matter who is inserting. When you pass from one tissue to another one, you also can detect this. You cannot, and this is true, with a robot, you can say this is tissue 1, this is tissue 2. So this is fat, this is muscle. When you have a human, it's more difficult. But the border between both always will be separated because we will have an abrupt change in the dynamic of the signal. And this always is identifiable, doesn't matter who is inserting. Yeah.
Thank you. Jürgen Drescher, there was another question.
Dr. Alfredo, congratulations. We have to meet in the happy hour because we have a very powerful robotic department in Oberpfaffenhofen nearby Munich. and we developed an active feedback loop robot. Actually, in the United States, company Medtronic uses it for very, very tiny operations, but your technology is not involved yet. But one thing I want to mention, there was a space shuttle mission STS-90 with micro in 1999. I was a research scientist there, and we did microneurography the same way like you did via audio guidance to find a nerve in a tissue to measure sympathetic nervous activity. So we should discuss this later, and we have excellent overlapping. And with your technology, you can be not only in terrestrial medicine, also in space medicine. You will find a lot of applications.
So thank you very much. So great, yeah. So we can meet in the booth if you want, and I will be very interested to read this paper about. I would like to know which kind of sensor they are using.
Yes. Okay.
Thank you very much.
Thank you as well. I had just one question, because I realized that you work also between academia and the industry, which is very similar to Fraunhofer. So I would like to know, how is your experience in working with the industry, and which challenges do you face when it comes to create a first contact? Because I think this is always, we had this topic yesterday as well, it's a cultural aspect between institutions, and I would like to know how you face it.
I mean, the first problem is IPs. I think that, yeah, so I am employee of the university, but through this Exist Research Transfer Project, we have funded a company. So SURAG today is a startup that was created on August. I think that the IP issues are very complex. Yeah, we need to. I mean, we have to do a negotiation that is quite long. I think that both sides. I work for both sides. So I am an employee from the university since 6 years. I am CEO of this startup. And I think that something very important is that the rules have to be set at the beginning, and this could be done only about a clear, transparent, we will say, strategy, IP strategy from the university side. Yeah, when this is clear, what are the paths, and we know what a guideline about that is very clear. What are the steps to follow? Yeah, I have the same problem in Chile. In Chile also, I was assistant professor at one university, and then also I create a startup, and also the IP was an issue. Yeah, so I think that this is in our experience, this is the more important issue that should be solved, and we should think in a really early stage.
Okay.
Yeah.
Thank you for that. Yes, for the others that maybe don't know, of course, INKA created a startup, as you just mentioned, and we're going to hear a presentation from this startup tomorrow by Rutuja. And actually, the startup and the University of Magdeburg were part of the tandems that traveled to the US in June. So yeah, we're happy that you're here again. So thank you so much, Alfredo. And if there are more questions and you want to discuss, go to the booth, to the INKA booth afterwards in the exhibition hall. And now we are moving a little bit more to...